Okay. So the malaria endemic setting that I've been working in is Oceania, which is otherwise known as the Southwest Pacific, down here on the right, just above Australia. So that includes Papua New Guinea, Solomon Islands and Vanuatu. So in the Southwest Pacific, in Papua New Guinea, we have a human population size of around 7.5 million people. We have 5 different Plasmodium species, so the only one that isn't there is Plasmodium knowlesi. Falciparum's dominant in most areas, but now as transmission's going down, we're also seeing vivax becoming more dominant. We see transmission, as you can see from this endemicity map, in the lowlands and the coastal regions, and there's a very large mountain range right through the middle of, from west to east in Papua New Guinea, and there's basically epidemics and some endemic vivax in the highlands, and there's no malaria above 1,700 metres. In the Solomon Islands, the human population size is much smaller. It's about 600,000 people. There's 9 large island provinces, so the population is quite dense in the middle islands, and you can see that's where there's higher transmission. But in the Solomon Islands, there's sort of low to moderate transmission, whereas in Papua New Guinea it's considered high. Vivax is dominant in the community, and it's also highly heterogeneous across different islands. So both Papua New Guinea and the Solomon Islands have adopted a goal of malaria elimination by 2030. It's a quite ambitious goal, but they're aiming for that, along with other countries in the Asia Pacific. So in Papua New Guinea, it scaled up its control program relatively late, and since about 2006, and that was through funding from the Global Fund, which allowed the 2 rollouts of bed nets in 2004 and 2009. So the 2004 rollout was completed in about 2008. And in 2012, there was a switch to artemisinin and combination therapies and a test and treat policy. So since 2010, we've seen a 75% decline in cases and deaths. And if you go into the community and look at the PCR prevalence, we've seen large declines in both Plasmodium falciparum and Plasmodium vivax. Though in Madang, on the right here in 2014, we have seen vivax coming back, and that work's now published. So in about 2010, we published a paper describing the population structure of falciparum in Papua New Guinea, and this was based on samples just from the north coast of Papua New Guinea. On the north coast of Papua New Guinea here, and we found quite strong population structure based on microsatellite markers. And so this then sort of provided the impetus for the work that I'm going to tell you about today. But I just wanted to show you this plot, which includes some further data points from 2010 and 2014 in Madang, and from 2013 in East Sepik. So you can see that here the populations look quite different to the other time points, and that's because that data set, we didn't have the raw data. So we couldn't actually reanalyse the raw microsatellite data together, and that's quite important for microsatellites. It's very hard to combine different data sets if you don't have the raw data, and that's because the allele calling is very specific to a particular data set, and sometimes even the machine that you've run the genotyping on. So we were looking to develop some new genotyping approaches, and we were considering that they needed to be reproducible and flexible, and we needed to be able to transfer these genotyping approaches to endemic countries for in-country surveillance. And microsatellites weren't really amenable to that goal because we have to send them away to a sequencing facility. But SNP barcodes, for example, with a biplex machine, which we had in country in Papua New Guinea, we could potentially develop SNP barcodes. So also what we had to think about here was there was significant global population structure, and Papua New Guinea really stuck out like a sore thumb. It looks quite different to other parasite populations. And so the global barcode that had been developed, although very useful for many different questions, we thought may not actually provide the resolution we needed for the spatial scale relevant to us and to the PNG and Solomon Islands national control programs. So we decided to develop a SNP barcode for Pacific Plasmodium falciparum, and we did this by sequencing parasite isolates from 3 different areas of PNG. They were clinical isolates. We sent them to the Sangha, and they were sequenced as part of the Malaria Gen Community Project. We ended up with, from about 150 isolates, about 91 high-quality genotypes. Genotyped against the Malaria Gen data set. There's now 1 million SNPs, but we had 600. The set that we compared our samples against were, at that stage, there were about 600,000 SNPs. Of those, about 24,000 were polymorphic in PNG. And then we selected SNPs that had a reasonably moderate to high minor allele frequency of at least 10%. And then so what we did then was we did some analyses. We measured FST for each SNP, so we were able to rank them by their FST value. We also identified those that were in neutral regions of the genome, so we measured selection across the genome and we chose SNPs outside regions under selection. And we also did logistic regression to identify SNPs that could discriminate between the 3 populations as well. So we ended up choosing 192 SNPs for our assay, and we developed a fluidigm-based assay to genotype our parasites. So just before I move on, I just want to show you how different sets of SNPs can give you different patterns of population structure. So here we have all of the PNG SNPs, and you can see that the 3 populations cluster separately, relatively separately, although there is still some overlap. This is 100 SNPs that Sarah Volkman provided to me that were based on moderate allele frequencies in different global populations. So they don't discriminate between our PNG parasite populations very well, whereas our Pacific barcode does discriminate between our populations. And then if we split the barcode into the SNPs with the FST higher than 0.05 and those that have less than 0.05, you can see the different patterns that we get. So you can clearly see that different SNPs give you different patterns, but what we decided to do was to genotype our parasites with the entire barcode because we felt that that was giving us, a reasonable set of discrimination, and then depending on what we wanted to use those SNPs for, we could use the subsets of SNPs. So we then applied the barcode to the PNG National Malaria Indicator Survey. This involved collection of 10,000 samples from across the country in 17 provinces. These are the malaria endemic provinces. All of the samples were species typed to identify the infections, and we determined multiplicity of infection using PFMSP2 for the falciparum samples, and we've also done this on Vivax as well. So then we took the samples that had an MOI of 1 or 2, and we applied the barcode to those samples. So this is a summary of the genetic diversity in different populations across PNG, and the colours in the bars are consistent with those on the map. So we can see that there's high diversity pretty much across the whole country, except this one population here from the south coast. This is over here. And some of these populations here have quite high diversity, but that's representative of just a few samples. So basically what we suspected here was that on the south coast, we knew that there was very low transmission. And in these populations here, there's a few quite diverse parasites. It's possible that they've been imported from other parts of the country. So when we looked at the population structure, we just did some multi-dimensional scaling, which is similar to principal components analysis, except it allows for missing data. So basically there seems to be a big cluster, however there's a bit of a gradient of population structure in the samples. We have some outliers as well, as you can see here. And this is the structure analysis. So this is a Bayesian clustering method that divides the data into genetic clusters, and each of the bars represents a different genotype and the membership or the ancestry of that genotype in a particular genetic cluster. So we can see that there's population structure that's confirming our earlier microsatellite data, and we see that there's some populations that are quite isolated or genetically isolated, and this island population over here seems to be quite different to the rest of PNG. So these are from across the north coast, and then here we have this population here in Milne Bay, and this population here from the islands. So basically this data enables us to define regional blocks, potentially for malaria elimination, and it helps us to understand the transmission scenario in different geographic regions. So we have these regional blocks in the Sepik, Madang, Morobe region and the islands. And we have this relatively isolated population over here, although we didn't have many samples from this region here, so we can't really tell whether there might be sort of a further gradient here. So we would need more samples from that region. So here, this is Manus Island. That's another distinct parasite population. And this is the Morobe region. So that's made up of 3 different types of genetic clusters of parasites. So some that appear to have been imported from the islands as well. And then we have clusters of imported parasites. So over here in the Sepik, this cluster here, they were related to Indonesian parasites, so possibly an imported cluster of parasites. They're also coming from the same village. So basically we think that parasites come from Indonesia and then spread through the population. And we also have on the south coast, as I mentioned earlier, this confirmed our suspicion that parasites were imported from multiple regions, and potentially in some parts of the south coast malaria may have, or falciparum may have already been eliminated. So here, this really demonstrates how we can classify the transmission scenario in different regions and regional blocks as units for elimination. Now this data was, the samples that were collected in 2008, and we've seen a lot of changes since then. So now we'd like to apply this barcode to more recent samples. In addition, we've used the barcode to genotype samples from migrating workers working in a resource company in Papua New Guinea, and they turn out to be form 2 clusters. We did a phylogenetic analysis, so we see this sort of imported parasite that's caused a local outbreak, and then we have other imported infections that are related to parasites from other populations, so directly imported, so migrating workers coming back from their endemic, from their home province and bringing parasites back to the camps. In the Solomon Islands, they've had 20 years of sustained control. So malaria has come down quite a lot. There's quite low transmission. Vivax now causes 50% of all clinical cases. It's more prevalent in the community, and there's now pilot elimination programs happening in the Solomon Islands. So in the Solomon Islands, we see a slightly different scenario. We didn't have as many falciparum samples because there's just not as much there. So we had samples from this island of Gela in Central Province, and also some samples from Guadalcanal, which is home to the capital city, and Malata. So basically, when we drew the multidimensional scaling plot, we could see that the parasite populations were quite distinct, and Gela formed quite an even more distinct and inbred cluster. And the Tetere parasites, the blue parasites over here, formed two different groups. So there was this really clonal cluster that clustered with the Geller parasites, and this more diverse set of parasites, which we believe is really the local parasite population, and the relatively clonal cluster was probably an imported parasite from Geller that's spread through the community. So just because I thought people might be interested in this, the connections between PNG and Solomon Islands are quite interesting, because here's the PNG parasites over here, and the Solomon Islands parasites tend to cluster with this parasite population over here. So there seems to be more connections over sea than over land. So these parasites from Milne Bay are more related to Solomon Islands parasites. So, malaria has no borders. So this is really important to remember this in malaria elimination. So now what we're doing is we're developing these SNP barcodes for rapid surveillance and outbreak mapping. So we want to transfer them to Papua New Guinea. We're trying to identify the best SNPs for these out of our barcode for this goal. So how do we actually do that? If you're wanting to define the minimum number of SNPs, you need to look at the number of SNPs in the haplotype and look at how well that defines your interpopulation differentiation or your population structure. FST is quite a traditional statistic, so it would be good to know whether that would be useful. But whoops, what have I done there? Turned it off. So we've also done an analysis method called multi-class linear discriminant analysis. And basically what this does is it identifies the SNPs that discriminate best between your parasite populations. So we've done this 6 different times, and then we've identified six different clustering patterns, and then we've identified the top SNPs that best discriminate between our parasite populations. So then when we look at how well how accurately those SNPs can distinguish between parasites from different populations, we find that actually our, if you look here, so this is the Pacific barcode over here with the SNPs that have the high FST, and so there's about 70 of those, and they can discriminate between our parasite populations just as well as those that we've identified through the linear discriminant analysis or through machine learning. And so really, if we use the whole barcode, we get much better discrimination capacity. And so there's really, as you can see, a trade-off between barcode size and accuracy. So just to summarise, local SNP barcodes are informing on malaria control on a range of spatial scales in Papua New Guinea and Solomon Islands. We can define transmission dynamics and networks. We can also define fragmented populations or regional blocks as potential units for elimination. We can trace origins of parasites, determining whether they're local or imported infections, and also looking at their geographic provenance. But the choice of the markers is really critical. It depends on the question, and the resolution and accuracy is really dependent on the barcode size and discrimination capacity. And ranking SNPs by FST seems to be just as effective as other more sophisticated methods to identify a minimal barcode. However, it really is the size quality kind of trade-off. And here are my acknowledgements. So I'd like to thank all my collaborators and my lab at WeHi and the funders. Okay, thank you. Now we have 40 seconds for questions.